What is gene therapy? Gene therapy is a test method that employs qualities to treat or avoid disease. Within the future, this strategy may permit specialists to treat a clutter by inserting a gene into a patient's cells rather than utilizing drugs or surgery. Researchers are testing a few approaches to quality treatment, including replacing a mutated gene that causes disease with a healthy duplicate of the gene, inactivating or knocking out a transformed gene that is functioning improperly, and lastly, introducing a new gene into the body to help fight the disease. There are two different types of gene therapy depending on which sorts of cells are treated. Somatic gene therapy, transfer of a segment of DNA to any cell of the body that doesn't deliver sperm or eggs. Impacts of gene therapy not be passed onto the patient's children. Germline gene therapy, transfer of a segment of DNA to cells that deliver eggs or sperm. Impacts of quality treatment will be passed onto the patient's children and consequent generations. What is cell therapy? Cell therapy aims to introduce new healthy cells into a patient's body to replace the infected or missing ones. A challenge for this sort of treatment is having sufficient cells for transplantation into a patient. This is because specialized cells such as brain cells are difficult to get from the human body. Also, particular cells regularly have a restricted capacity to increase, making it difficult to deliver sufficient numbers of cells required for certain cell treatments. A few of these issues can be overcome through the utilization of stem cells.
How does gene therapy work? Gene therapy is designed to introduce genetic material into cells to compensate for abnormal gene or to make beneficial protein. If the mutated gene causes necessary protein to be faulty or missing, gene therapy may be able to introduce a normal copy of the gene to restore the function of the protein. A gene that is inserted directly into the cell usually does not function. Instead, a carrier called vector is genetically engineered to deliver the gene. Certain viruses are often used as vector because they can deliver the new gene by infecting the cell. Then, what are the strategies being used in gene therapy? The gene therapy is a broad one. It encompasses many different strategies, all of which are designed to overcome or to alleviate this disease by a procedure in which gene segments or oligonucleotides are introduced into a cell of many affected individuals. The genetic material may be transferred directly into cell within a patient, or a cell may be removed from the patient and the genetic material inserted to them in vitro prior to replacing the cell in the patient.
So what exactly are the uses of gene therapy? For me, the main goal of gene therapy is to make advancement in the field of medicine. Gene therapy are commonly used to replace genes that are defective or missing to help cure certain diseases. They can also be used to deliver bacterial or viral genes as a form of a vaccine to cure or treat a disease faster. So they can also speed up the destruction of cancer cells or supply genes that could cause cancer cells to revert back to regular ones, therefore treating them. They are also developing genes that will stimulate healing faster and promote or stop the growth of its tissue. There are a lot of cell therapies being developed right now. The most common type, however, is blood transfusion, where red and white blood cells as well as platelets are transfused to an individual from donor. Another one is transplantation of hematopoietic stem cells to create bone marrow. Here are some examples of type of cells used for cell therapy. There are 4 types of pluripotent stem cells, which are the embryonic stem cells, induced pluripotent stem cells, nuclear transfer embryonic stem cells, and parthenogenetic embryonic stem cells. And there are also 4 types of multipotent stem cells, which were obtained from adult tissues, called hematopoietic stem cells, mesenchymal stem cells, neural stem cells, and epithelial stem cells.
Can viral vectors make you sick? The answer is that is no, because viral vectors vaccine use a modified version of a virus that is different from the virus being targeted to deliver important instructions to our cells. The modified version of the virus is called vector virus. Like all vaccines, viral vectors vaccine benefits people who get vaccinated by giving them protection against the disease like COVID-19 without them having to risk the serious consequences of getting sick. This virus protect your immune system by promoting your immune system to produce virus-fighting antibodies. How are viruses used in gene therapy? The most common gene therapy is they are using viral vectors, virus mostly, because viral vectors can be used for delivered functional genes to replace defective genes to cure genetic disorders. As a vaccine platform, viral vectors can be used for expression and the presentation of pathogenic antigens to induce an immune response by mimicking natural infections. Certain viruses are often used as a vectors because they can deliver a new gene by infecting the cell.